23.3 groups of protists. By the end of this section, you will be able to do the following. Describe representative protist organisms from each of the 6 presently recognized supergroups of eukaryotes. Identify the evolutionary relationships of plants, animals, and fungi within the 6 presently recognized supergroups of eukaryotes. Identify defining features of protists in each of the 6 supergroups of eukaryotes. In the span of several decades, the kingdom Protista has been disassembled because sequence analyses have revealed new genetic, and therefore evolutionary, relationships among these eukaryotes. Moreover, protists that exhibit similar morphological features may have evolved analogous structures because of similar selective pressures, rather than because of 582 Chapter 23. Protists access for free at openstax.org. Recent common ancestry. This phenomenon, called convergent evolution, is one reason why protist classification is so challenging. The emerging classification scheme groups the entire domain Eukarya into 6 supergroups that contain all of the protists as well as animals, plants, and fungi that evolved from a common ancestor, figure 23.9. Each of the supergroups is believed to be monophyletic, meaning that all organisms within each supergroup are believed to have evolved from a single common ancestor, and thus all members are most closely related to each other than to organisms outside that group. There is still evidence lacking for the monophily of some groups. Each supergroup can be viewed as representing one of many variants on eukaryotic cell structure. In each group, one or more of the defining characters of the eukaryotic cell, the nucleus, the cytoskeleton, and the endosymbiotic organelles, may have diverged from the typical pattern. Figure 23.9 Eukaryotic supergroups. This diagram shows a proposed classification of the domain Eukarya. Currently, the domain Eukarya is divided into 6 supergroups. Within each supergroup are multiple kingdoms. Although each supergroup is believed to be monophyletic, the dotted lines suggest evolutionary relationships among the supergroups that continue to be debated. 23.3. Groups of protists 583. Keep in mind that the classification scheme presented here represents just one of several hypotheses, and the true evolutionary relationships are still to be determined. The 6 supergroups may be modified or replaced by a more appropriate hierarchy as genetic, morphological, and ecological data accumulate. When learning about protists, it is helpful to focus less on the nomenclature and more on the commonalities and differences that illustrate how each group has exploited the possibilities of eukaryotic life. Archaeplastida molecular evidence supports the hypothesis that all Archaeplastida are descendants of an endosymbiotic relationship between a heterotrophic protist and a cyanobacterium. The protist members of the group include the red algae and green algae. It was from a common ancestor of these protists that the land plants evolved, since their closest relatives are found in this group. The red and green algae include unicellular, multicellular, and colonial forms. A variety of algal life cycles exists, but the most complex is alternation of generations, in which both haploid and diploid stages are multicellular. A diploid sporophyte contains cells that undergo meiosis to produce haploid spores. The spores germinate and grow into a haploid gametophyte, which then makes gametes by mitosis. The gametes fuse to form a zygote that grows into a diploid sporophyte. Alternation of generations is seen in some species of archaeplastid algae, as well as some species of stramenopiles, figure 23.10. In some species, the gametophyte and sporophyte look quite different, while in others they are nearly indistinguishable. Glaucophytes are a small group of archaeplastida interesting because their chloroplasts retain remnants of the peptidoglycan cell wall of the ancestral cyanobacterial endosymbiont, figure 23.10. Figure 23.10 Glaucocystis. Credit. By Ya. User. Neon Commons. User. Neon_Ya. Own work. CC by SA 2.5. https://commons.wikimedia.org/w/index.php. Current equals 1706641. http://openstax.org/l/glaucocystis closing parenthesis. Red algae, or rhodophytes lack flagella, and are primarily multicellular, although they range in size from microscopic, unicellular protists to large, multicellular forms grouped into the informal seaweed category. Red algae have a second cell wall outside an inner cellulose cell wall. Carbohydrates in this wall are the source of agarose used for electrophoresis gels and agar for solidifying bacterial media. The red in the red algae comes from phycoerythrins, accessory photopigments that are red in color and obscure the green tint of chlorophyll in some species. Other protists classified as red algae lack phycoerythrins and are 584. Chapter 23. Protists access for free at openstax.org. Parasites. Both the red algae and the glaucophytes store carbohydrates in the cytoplasm rather than in the plastid. Red algae are common in tropical waters where they have been detected at depths of 260 meters. Other red algae exist in terrestrial or freshwater environments. The red algae life cycle is an unusual alternation of generations that includes two sporophyte phases, with meiosis occurring only in the second sporophyte. Green algae. Chlorophytes and charophytes. The most abundant group of algae is the green algae. The green algae exhibit features similar to those of the land plants, particularly in terms of chloroplast structure. In both green algae and plants, carbohydrates are stored in the plastid. That this group of protists shared a relatively recent common ancestor with land plants is well supported. The green algae are subdivided into the chlorophytes and the charophytes. The charophytes are the closest living relatives to land plants and resemble them in morphology and reproductive strategies. The familiar Spirogyra is a charophyte. Charophytes are common in wet habitats, and their presence often signals a healthy ecosystem. The chlorophytes exhibit great diversity of form and function. Chlorophytes primarily inhabit freshwater and damp soil, and are a common component of plankton. Chlamydomonas is a simple, unicellular chlorophyte with a pear-shaped morphology and two opposing, anterior flagella that guide this protist toward light sensed by its eye spot. More complex chlorophyte species exhibit haploid gametes and spores that resemble chlamydomonas. The chlorophyte volvox is one of only a few examples of a colonial organism, which behaves in some ways like a collection of individual cells, but in other ways like the specialized cells of a multicellular organism. Figure 23.11. Volvox colonies contain 500 to 60,000 cells, each with two flagella, contained within a hollow, spherical matrix composed of a gelatinous glycoprotein secretion. Individual cells in a Volvox colony move in a coordinated fashion and are interconnected by cytoplasmic bridges. Only a few of the cells reproduce to create daughter colonies, an example of basic cell specialization in this organism. Daughter colonies are produced with their flagella on the inside and have to evert as they are released. Figure 23.11 Volvox. Volvox aureus is a green alga in the supergroup Archaeplastida. This species exists as a colony, consisting of cells immersed in a gel-like matrix and intertwined with each other via hair-like cytoplasmic extensions. Credit: Dr. Ralph Wagner. True multicellular organisms, such as the sea lettuce, ulva, are also represented among the chlorophytes. In addition, some chlorophytes exist as large, multinucleate, single cells. Species in the genus Collarpaw exhibit flattened fern-like foliage and can reach lengths of 3 meters. Figure 23.12. Collarpaw species undergo nuclear division, but their cells do not complete cytokinesis, remaining instead as massive and elaborate single cells. 23.3. Groups of protists 585. Figure 23.12. A multinucleate alga. Collarpaw taxifolia is a chlorophyte consisting of a single cell containing potentially thousands of nuclei. Credit: Noah. An interesting question is how a single cell can produce such complex shapes. Link to learning. Take a look at this video to see cytoplasmic streaming in a green alga. Click to view content, https://www.openstax.org/l/chara_corallina. Amoebozoa. Like the Archaeplastida, the amoebozoa include species with single cells, species with large multinucleated cells, and species that have multicellular phases. Amoebozoan cells characteristically exhibit pseudopodia that extend like tubes or flat lobes. These pseudopods project outward from anywhere on the cell surface and can anchor to a substrate. The protist then transports its cytoplasm into the pseudopod, thereby moving the entire cell. This type of motion is similar to the cytoplasmic streaming used to move organelles in the Archaeplastida, and is also used by other protists as a means of locomotion or as a method to distribute nutrients and oxygen. The amoebozoa include both free-living and parasitic species. Gymnamoebae. The gymnamoeba or lobose amoebae include both naked amoebae like the familiar amoeba Proteus and shelled amoebae, whose bodies protrude like snails from their protective tests. Amoeba Proteus is a large amoeba about 500 micrometers in diameter but is dwarfed by the multinucleate amoebae Polomixa, which can be 10 times its size. Although Polomixa may have hundreds of nuclei, it has lost its mitochondria, but replaced them with bacterial endosymbionts. The secondary loss or modification of mitochondria is a feature also seen in other protist groups. Figure 23.13 Amoeba. Amoebae with tubular and lobe-shaped pseudopodia are seen under a microscope. These isolates would be morphologically classified as amoebozoans. 586 Chapter 23. Protists access for free at openstax.org. Slime molds. A subset of the amoebozoans, the slime molds, has several morphological similarities to fungi that are thought to be the result of convergent evolution. For instance, during times of stress, some slime molds develop into spore-generating fruiting bodies, much like fungi. The slime molds are categorized on the basis of their life cycles into plasmodial or cellular types. Plasmodial slime molds are composed of large, multinucleate cells and move along surfaces like an amorphous blob of slime during their feeding stage. Figure 23.14. Food particles are lifted and engulfed into the slime mold as it glides along. The dog vomit slime mold seen in Figure 23.14 is a particularly colorful specimen, and its ability to creep about might well trigger suspicion of alien invasion. Upon maturation, the plasmodium takes on a net-like appearance with the ability to form fruiting bodies, or sporangia, during times of stress. Haploid spores are produced by meiosis within the sporangia, and spores can be disseminated through the air or water to potentially land in more favorable environments. If this occurs, the spores germinate to form amoeboid or flagellate haploid cells that can combine with each other and produce a diploid zygotic slime mold to complete the life cycle. Figure 23.14 Plasmodial slime molds. The life cycle of the plasmodial slime mold is shown. The brightly colored plasmodium in the inset photo is a single-celled, multinucleate mass. Credit. Modification of work by Dr. Jonathan Gott and the Center for RNA Molecular Biology, Case Western Reserve University. The cellular slime molds function as independent amoeboid cells when nutrients are abundant. When food is depleted, cellular slime molds aggregate into a mass of cells that behaves as a single unit, called a slug. Some cells in the slug contribute to a 2 to 3 mm stalk, drying up and dying in the process. Cells atop the stalk form an asexual fruiting body that contains haploid spores, Figure 23.15. As with plasmodial slime molds, the spores are disseminated and can germinate if they land in a moist environment. One representative genus of the cellular slime molds is Dictyostelium, which commonly exists in the damp 23.3. Groups of protists 587 soil of forests. Figure 23.15 Cellular slime mold. The image shows several stages in the life cycle of Dictyostelium discoideum, including aggregated cells, mobile slugs and their transformation into fruiting bodies with a cluster of spores supported by a stalk. Credit. By Usman Bashir, own work, CC by SAW 4.0. http://creativecommons.org/licenses/by-saw/4.0 http://openstax.org/l/ccb. Via Wikimedia Commons. Link to learning. View this video to see the formation of a fruiting body by a cellular slime mold. Click to view content, https://www.openstax.org/l/slime_mold. Opistheconta. The opisthecants are named for the single posterior flagellum seen in flagellated cells of the group. The flagella of other protists are anterior and their movement pulls the cells along, while the opisthocons are pushed. Protist members of the opisthocons include the animal-like choanoflagellates, which are believed to resemble the common ancestor of sponges and perhaps all animals. Choanoflagellates include unicellular and colonial forms, Figure 23.16, and number about 244 described species. In these organisms, the single, apical flagellum is surrounded by a contractile collar composed of microvilli. The collar is used to filter and collect bacteria for ingestion by the protist. A similar feeding mechanism is seen in the collar cells of sponges, which suggests a possible connection between choanoflagellates and animals. The Mesomycetozoa form a small group of parasites, primarily of fish, and at least one form that can parasitize humans. Their life cycles are poorly understood. These organisms are of special interest, because they appear to be so closely related to animals. In the past, they were grouped with fungi and other protists based on their morphology. 588 Chapter 23, Protists Access for free at openstax.org. Figure 23.16 A colonial choanoflagellate. Credit. By D. H. Jeanette, http://en.wikipedia.org/wiki/choanoflagellate, http://openstax.org/l/choano, public domain. Via Wikimedia Commons. The previous supergroups are all the products of primary endosymbiontic events and their organelles, nucleus, mitochondria, and chloroplasts, or what would be considered typical, i.e., matching the diagrams you would find in an introductory biology book. The next three supergroups all contain at least some photosynthetic members whose chloroplasts were derived by secondary endosymbiosis. They also show some interesting variations in nuclear structure and modification of mitochondria or chloroplasts. Rosaria. The Rosaria supergroup includes many of the amoebas with thin thread-like, needle-like or root-like pseudopodia, figure 23.17, rather than the broader lobed pseudopodia of the amoebozoa. Many rhizarians make elaborate and beautiful tests, armor-like coverings for the body of the cell, composed of calcium carbonate, silicon, or strontium salts. Rhizarians have important roles in both carbon and nitrogen cycles. When rhizarians die, and their tests sink into deep water, the carbonates are out of reach of most decomposers, locking carbon dioxide away from the atmosphere. In general, this process by which carbon is transported deep into the ocean is described as the biological carbon pump, because carbon is pumped to the ocean depths where it is inaccessible to the atmosphere as carbon dioxide. The biological carbon pump is a crucial component of the carbon cycle that maintains lower atmospheric carbon dioxide levels. Foraminiferans are unusual in that they are the only eukaryotes known to participate in the nitrogen cycle by denitrification, an activity usually served only by prokaryotes. Figure 23.17 Rosaria. Ammonia tepida, a Rosaria species viewed here using phase contrast light microscopy, exhibits many thread-like 23.3 groups of protists 589 pseudopodia. It also has a chambered calcium carbonate shell or test. Credit. Modification of work by Scott Fay, UC Berkeley. Scale bar data from Matt Russell. Foraminiferans, or forams, are unicellular heterotrophic protists, ranging from approximately 20 micrometers to several centimeters in length, and occasionally resembling tiny snails, figure 23.18. As a group, the forams exhibit porous shells, called tests that are built from various organic materials and typically hardened with calcium carbonate. The tests may house photosynthetic algae, which the forams can harvest for nutrition. Foram pseudopodia extend through the pores and allow the forams to move, feed, and gather additional building materials. Typically, forams are associated with sand or other particles in marine or freshwater habitats. Foraminiferans are also useful as indicators of pollution and changes in global weather patterns. Figure 23.18 Foraminiferan tests. These shells from foraminifera sank to the seafloor. Credit. Deep East 2001, NOAA, OER, Radiolarians. A second subtype of Rosaria, the radiolarians, exhibit intricate exteriors of glassy silica with radial or bilateral symmetry, figure 23.19. Needle-like pseudopods supported by microtubules radiate outward from the cell bodies of these protists and function to catch food particles. The shells of dead radiolarians sink to the ocean floor, where they may accumulate in 100-meter thick depths. Preserved, sedimented radiolarians are very common in the fossil record. Figure 23.19 Radiolarian shell. This fossilized radiolarian shell was imaged using a scanning electron microscope. Credit. Modification of work by Hannes Grobe, Alfred Wegener Institute. Scale bar data from Matt Russell. Cercozoa. The cercozoa are both morphologically and metabolically diverse, and include both naked and shelled forms. The chlorarachniophytes, figure 23.20, are photosynthetic, having acquired chloroplasts by secondary endosymbiosis. The 590 Chapter 23, Protists access for free at openstax.org. Chloroplast contains a remnant of the chlorophyte endosymbiont nucleus, sandwiched between the two sets of chloroplast membranes. Vampirellids or vampire amoebae, as their name suggests, obtain their nutrients by thrusting a pseudopod into the interior of other cells and sucking out their contents. Figure 23.20a chlorarachniophyte. This rhizarian is mixotrophic, and can obtain nutrients both by photosynthesis and by trapping various microorganisms with its network of pseudopodia. Credit. By Ya. User. Neon Commons. User. Neon_Ya. Own work. CC by SA 2.5. http://creativecommons.org/licenses/by-SA/2.5. http://openstax.org/l/ccb_25 or CC by SA 2.5. http://creativecommons.org/licenses/by-SA 2.5. http://openstax.org/l/ccb_25 Via Wikimedia Commons, chromalveolata. Current evidence suggests that species classified as chromalveolates are derived from a common ancestor that engulfed a photosynthetic red algal cell, which itself had already evolved chloroplasts from an endosymbiotic relationship with a photosynthetic prokaryote. Therefore, the ancestor of chromalveolates is believed to have resulted from a secondary endosymbiotic event. However, some chromalveolates appear to have lost red alga-derived plastid organelles or lack plastid genes altogether. Therefore, this supergroup should be considered a hypothesis-based working group that is subject to change. Chromalveolates include very important photosynthetic organisms, such as diatoms, brown algae, and significant disease agents in animals and plants. The chromalveolates can be subdivided into alveolates and strumenophiles. Alveolates, dinoflagellates, apicomplexians, and ciliates. A large body of data supports that the alveolates are derived from a shared common ancestor. The alveolates are named for the presence of an alveolus, or membrane-enclosed sac, beneath the cell membrane. The exact function of the alveolus is unknown, but it may be involved in osmoregulation. The alveolates are further categorized into some of the better-known protists: the dinoflagellates, the apicomplexans, and the ciliates. Dinoflagellates exhibit extensive morphological diversity and can be photosynthetic, heterotrophic, or mixotrophic. The chloroplast of photosynthetic dinoflagellates was derived by secondary endosymbiosis of a red alga. Many dinoflagellates are encased in interlocking plates of cellulose. 2 perpendicular flagella fit into the grooves between the cellulose plates, with one flagellum extending longitudinally and a second encircling the dinoflagellate. Figure 23.21. Together, the flagella contribute to the characteristic spinning motion of dinoflagellates. These protists exist in freshwater and marine habitats, and are a component of plankton, the typically microscopic organisms that drift through the water and serve as a crucial food source for larger aquatic organisms. 23.3. Groups of protists. 591. Figure 23.21. Dinoflagellates. The dinoflagellates exhibit great diversity in shape. Many are encased in cellulose armor and have two flagella that fit in grooves between the plates. Movement of these two perpendicular flagella causes a spinning motion. Dinoflagellates have a nuclear variant called a dinokaryon. The chromosomes in the dinokaryon are highly condensed throughout the cell cycle and do not have typical histones. Mitosis in dinoflagellates is closed. That is, the spindle separates the chromosomes from outside of the nucleus without breakdown of the nuclear envelope. Some dinoflagellates generate light, called bioluminescence, when they are jarred or stressed. Large numbers of marine dinoflagellates, billions or trillions of cells per wave, can emit light and cause an entire breaking wave to twinkle or take on a brilliant blue color. Figure 23.22. For approximately 20 species of marine dinoflagellates, population explosions, also called blooms, during the summer months can tint the ocean with a muddy red color. This phenomenon is called a red tide, and it results from the abundant red pigments present in dinoflagellate plastids. In large quantities, these dinoflagellate species secrete an asphyxiating toxin that can kill fish, birds, and marine mammals. Red tides can be massively detrimental to commercial fisheries, and humans who consume these protists may become poisoned. Figure 23.22 Dinoflagellate bioluminescence. Bioluminescence is emitted from dinoflagellates in a breaking wave, as seen from the New Jersey coast. Credit. Catalano 82 inches, Flickr. The apicomplexan protists are named for a structure called an apical complex, figure 23.23, which appears to be a highly modified secondary chloroplast. The apicoplast genome is similar to those of dinoflagellate chloroplasts. The apical complex is specialized for entry and infection of host cells. Indeed, all apicomplexans are parasitic. This group includes the genus Plasmodium, which causes malaria in humans. Apicomplexan life cycles are complex, involving multiple hosts and stages of sexual and asexual reproduction. 592 Chapter 23, Protists access for free at openstax.org. Figure 23.23 Apicomplexa. A. Apicomplexans are parasitic protists. They have a characteristic apical complex that enables them to infect host cells. B. Plasmodium, the causative agent of malaria, has a complex life cycle typical of apicomplexans. Credit B. Modification of work by CDC. The ciliates, which include Paramecium and Tetrahymena, are a group of protists 10 to 3000 micrometers in length that are covered in rows, tufts, or spirals of tiny cilia. By beating their cilia synchronously or in waves, ciliates can coordinate directed movements and ingest food particles. Certain ciliates have fused cilia-based structures that function like paddles, funnels, or fins. Ciliates also are surrounded by a pellicle, providing protection without compromising agility. The genus Paramecium includes protists that have organized their cilia into a plate-like primitive mouth, called an oral groove, which is used to capture and digest bacteria, figure 23.24. Food captured in the oral groove enters a food vacuole, where it combines with digestive enzymes. Waste particles are expelled by an exocytic vesicle that fuses at a specific region on the cell membrane, called the anal pore. In addition to a vacuole-based digestive system, Paramecium also uses contractile vacuoles, which are osmoregulatory vesicles that fill with water as it enters the cell by osmosis and then contract to squeeze water from the cell. Ciliates therefore exhibit considerable structural complexity without having achieved multicellularity. Figure 23.24 Paramecium. Paramecium has a primitive mouth, called an oral groove, to ingest food, and an anal pore to eliminate waste. Contractile vacuoles allow the organism to excrete excess water. Cilia enable the organism to move. Credit, Paramecium micrograph. Modification of work by NIH. Scale bar data from Matt Russell. Link to learning. Watch the video of the contractile vacuole of Paramecium expelling water to keep the cell osmotically balanced. Click to view content, https://www.openstax.org/l/paramecium. Paramecium has two nuclei, a macronucleus and a micronucleus, in each cell. The micronucleus is essential for sexual reproduction, and is in many ways a typical eukaryotic nucleus, except that its genes are not transcribed. The transcribed nucleus is the macronucleus, which directs asexual binary fission and all other biological functions. The macronucleus is a 23.3 groups of protists 593 multiploid nucleus constructed from the micronucleus during sexual reproduction. Periodic reconstruction of the macronucleus is necessary because the macronucleus divides amitotically, and thus becomes genetically unbalanced over a period of successive cell replications. Paramecium and most other ciliates reproduce sexually by conjugation. This process begins when two different mating types of Paramecium make physical contact and join with a cytoplasmic bridge, figure 23.25. The diploid micronucleus in each cell then undergoes meiosis to produce 4 haploid micronuclei. 3 of these degenerate in each cell, leaving 1 micronucleus that then undergoes mitosis, generating 2 haploid micronuclei. The cells each exchange one of these haploid nuclei and move away from each other. Fusion of the haploid micronuclei generates a completely novel diploid pre-micronucleus in each conjugative cell. This pre-micronucleus undergoes 3 rounds of mitosis to produce 8 copies, and the original macronucleus disintegrates. 4 of the 8 pre-micronuclei become full-fledged micronuclei, whereas the other 4 perform multiple rounds of DNA replication. The copies of the micronuclear chromosomes are severely edited to form hundreds of smaller chromosomes that contain only the protein-coding genes. Each of these smaller chromosomes gets new telomeres as the macronucleus differentiates. 2 cycles of cell division then yield 4 new paramecia from each original conjugative cell. Visual connection figure 23.25 Conjugation in paramecium. The complex process of sexual reproduction in paramecium creates 8 daughter cells from 2 original cells. Each cell has a macronucleus and a micronucleus. During sexual reproduction, the macronucleus dissolves and is 594 Chapter 23. Protists access for free at openstax.org. Replaced by a micronucleus. Credit, Micrograph. Modification of work by Ian Sutton. Scale bar data from Matt Russell. Which of the following statements about paramecium sexual reproduction is false? A. The macronuclei are derived from micronuclei. B. Both mitosis and meiosis occur during sexual reproduction. C. The conjugate pair swaps macronuclei. D. Each parent produces 4 daughter cells. Stramenophiles. Diatoms, brown algae, golden algae and oomycetes. The other subgroup of chromalveolates, the stramenophiles, includes photosynthetic marine algae and heterotrophic protists. The chloroplast of these algae is derived from red alga. The identifying feature of this group is the presence of a textured, or hairy, flagellum. Many stramenophiles also have an additional flagellum that lacks hair-like projections. Figure 23.26. Members of this subgroup range in size from single-celled diatoms to the massive and multicellular kelp. Figure 23.26 Stramenophile flagella. This stramenophile cell has a single hairy flagellum and a secondary smooth flagellum. The diatoms are unicellular photosynthetic protists that encase themselves in intricately patterned, glassy cell walls composed of silicon dioxide in a matrix of organic particles. Figure 23.27. These protists are a component of freshwater and marine plankton. Most species of diatoms reproduce asexually, although some instances of sexual reproduction and sporulation also exist. Some diatoms exhibit a slit in their silica shell, called a raphe. By expelling a stream of mucopolysaccharides from the raphe, the diatom can attach to surfaces or propel itself in one direction. 23.3. Groups of protists 595 Figure 23.27 Diatoms. Assorted diatoms, visualized here using light microscopy, live among annual sea ice in McMurdo Sound, Antarctica. Diatoms range in size from 2 to 200 micrometers. Credit. Professor Gordon T. Taylor, Stony Brook University, NSF, NOAA. During periods of nutrient availability, diatom populations bloom to numbers greater than can be consumed by aquatic organisms. The excess diatoms die and sink to the seafloor where they are not easily reached by assay probes that feed on dead organisms. As a result, the carbon dioxide that the diatoms had consumed and incorporated into their cells during photosynthesis is not returned to the atmosphere. Along with rhizarians and other shelled protists, diatoms help to maintain a balanced carbon cycle. Like diatoms, golden algae are largely unicellular, although some species can form large colonies. Their characteristic gold color results from their extensive use of carotenoids, a group of photosynthetic pigments that are generally yellow or orange in color. Golden algae are found in both freshwater and marine environments, where they form a major part of the plankton community. The brown algae are primarily marine, multicellular organisms that are known colloquially as seaweeds. Giant kelps are a type of brown alga. Some brown algae have evolved specialized tissues that resemble terrestrial plants, with root-like holdfasts, stem-like stipes, and leaf-like blades that are capable of photosynthesis. The stipes of giant kelps are enormous, extending in some cases for 60 meters. Like the green algae, brown algae have a variety of life cycles, including alternation of generations. In the brown algae genus Laminaria, haploid spores develop into multicellular gametophytes, which produce haploid gametes that combine to produce diploid organisms that then become multicellular organisms with a different structure from the haploid form. Figure 23.28. 596 Chapter 23, Protists access for free at OpenStax.org. Visual connection Figure 23.28 Alternation of generations in a brown alga. Several species of brown algae, such as the laminaria shown here, have evolved life cycles in which both the haploid, gametophyte, and diploid, sporophyte, forms are multicellular. The gametophyte is different in structure than the sporophyte. Credit, Laminaria photograph. Modification of work by Claire Fackler, CINMS, NOAA Photo Library. Which of the following statements about the laminaria life cycle is false? A. 1N zoospores form in the sporangia. B. The sporophyte is the 2N plant. C. The gametophyte is diploid. D. Both the gametophyte and sporophyte stages are multicellular. The water molds, oomycetes, egg fungus, were so named based on their fungus-like morphology, but molecular data have shown that the water molds are not closely related to fungi. The oomycetes are characterized by a cellulose-based cell wall and an extensive network of filaments that allow for nutrient uptake. As diploid spores, many oomycetes have two oppositely directed flagella, one hairy and one smooth, for locomotion. The oomycetes are non-photosynthetic and include many SA probes and parasites. The SA probes appear as white fluffy growths on dead organisms, Figure 23.29. Most oomycetes are aquatic, but some parasitize terrestrial plants. One plant pathogen is Phytophthora infestans, the causative agent of late blight of potatoes, such as occurred in the 19th century Irish potato famine. 23.3. Groups of protists 597 Figure 23.29 Oomycetes. A saprobic oomycete engulfs a dead insect. Credit. Modification of work by Thomas Bresson. Excavata. Many of the protist species classified into the supergroup Excavata are asymmetrical, single-celled organisms with a feeding groove, excavated from one side. This supergroup includes heterotrophic predators, photosynthetic species, and parasites. Its subgroups are the diplomonads, parabasalids, and euglenozoans. The group includes a variety of modified mitochondria, as well as chloroplasts derived from green algae by secondary endosymbiosis. Many of the euglenozoans are free-living, but most diplomonads and parabasalids are symbionts or parasites. Diplomonads among the Excavata are the diplomonads, which include the intestinal parasite, Giardia lamblia, Figure 23.30. Until recently, these protists were believed to lack mitochondria. Mitochondrial remnant organelles, called mitosomes, have since been identified in diplomonads, but although these mitosomes are essentially non-functional as respiratory organelles, they do function in iron and sulfur metabolism. Diplomonads exist in anaerobic environments and use alternative pathways, such as glycolysis, to generate energy. Each diplomonad cell has two similar, but not identical haploid nuclei. Diplomonads have four pairs of locomotor flagella that are fairly deeply rooted in basal bodies that lie between the two nuclei. Figure 23.30 Giardia. The mammalian intestinal parasite Giardia lamblia, visualized here using scanning electron microscopy, is a waterborne protist that causes severe diarrhea when ingested. Credit. Modification of work by Janice Carr, CDC. Scale bar data from Matt Russell, 598 Chapter 23, Protists access for free at OpenStax.org. Parabasalids. A second Excavata subgroup, the parabasalids, are named for the parabasal apparatus, which consists of a Golgi complex associated with cytoskeletal fibers. Other cytoskeletal features include an axostyle, a bundle of fibers that runs the length of the cell and may even extend beyond it. Parabasalids move with flagella and membrane rippling, and these and other cytoskeletal modifications may assist locomotion. Like the diplomonads, the parabasalids exhibit modified mitochondria. In parabasalids, these structures function anaerobically and are called hydrogenosomes because they produce hydrogen gas as a byproduct. The parabasalid Trichomonas vaginalis causes trichomoniasis, a sexually transmitted disease in humans, which appears in an estimated 180 million cases worldwide each year. Whereas men rarely exhibit symptoms during an infection with this protist, infected women may become more susceptible to secondary infection with human immunodeficiency virus, HIV, and may be more likely to develop cervical cancer. Pregnant women infected with T. vaginalis are at an increased risk of serious complications, such as preterm delivery. Some of the most complex of the parabasalids are those that colonize the rumen of ruminant animals and the guts of termites. These organisms can digest cellulose, a metabolic talent that is unusual among eukaryotic cells. They have multiple flagella arranged in complex patterns, and some additionally recruit spirochetes that attach to their surface to act as accessory locomotor structures. Link to learning termite gut endosymbionts. Click to view content, https://www.youtube.com/embed/hox7sdq. Euglenozoans includes parasites, heterotrophs, autotrophs, and mixotrophs, ranging in size from 10 to 500 micrometers. Euglenoids move through their aquatic habitats using two long flagella that guide them toward light sources sensed by a primitive ocular organ called an eyespot. The familiar genus, Euglena, encompasses some mixotrophic species that display a photosynthetic capability only when light is present. The chloroplast of Euglena descends from a green alga by secondary endosymbiosis. In the dark, the chloroplasts of Euglena shrink up and temporarily cease functioning, and the cells instead take up organic nutrients from their environment. Euglena has a tough pellicle composed of bands of protein attached to the cytoskeleton. The bands spiral around the cell and give Euglena its exceptional flexibility. The human parasite, Trypanosoma brucei, belongs to a different subgroup of Euglenozoa, the kind Toplastids. The kinetoplastid subgroup is named after the kinetoplast, a large modified mitochondrion carrying multiple circular DNAs. This subgroup includes several parasites, collectively called trypanosomes, which cause devastating human diseases and infect an insect species during a portion of their life cycle. T. brucei develops in the gut of the tsetse fly after the fly bites an infected human or other mammalian host. The parasite then travels to the insect salivary glands to be transmitted to another human or other mammal when the infected tsetse fly consumes another blood meal. T. brucei is common in Central Africa and is the causative agent of African sleeping sickness, a disease associated with severe chronic fatigue, coma, and can be fatal if left untreated since it leads to progressive decline of the function of the central nervous system. 23.3. Groups of protists. 599. Figure 23.31. Sleeping sickness. Trypanosoma brucei, the causative agent of sleeping sickness, spends part of its life cycle in the tsetse fly and part in humans. Credit. Modification of work by CDC. Link to learning. Watch this video to see T. brucei swimming. Click to view content, https://www.openstax.org/l/T_brucei.